Hello, respected judges of the Undergraduate Research and Innovation Competition. My name is Mallak Mansour, and together with my colleague Qamar Talal, we will be presenting our research titled Bone Marrow Derived Dendritic Cells Immunity Against Candida auris under the supervision of Dr. Mohammed Ghaleb. Imagine a silent invader so small that we cannot see, yet strong enough to shut down entire hospital units. That invader is Candida auris, a fungus unknown just 15 years ago, but now labeled by the CDC as a major global health threat. Its danger lies in its ability to persist on hospital surfaces, spread silently between patients, and resist multiple antifungal treatments. This graph demonstrates Candida auris infections have been increasing over the last decade in the United Arab Emirates. But while Candida auris is evolving, so is our immune system's ability to fight it. The dot plot shows that as time increases, the number of antigen presenting cells also increases. This means the immune response becomes stronger over time.
Before starting with our story, these illustrations were prepared by us using BioRender. The label given to this fungus by the CDC was never an overestimation, for when the Candida clan and its faction entered the body, what was once a peaceful kingdom turned into infiltration missions and marching control troops. Dendritic cells were the messengers, ready to inform the soldiers' T cells whenever threat lurked in human tissue. And Candida albicans was an intruder familiar to them, recognized by a distinct mark on their gears, beta-glucan, a structural mark embedded in their cell walls. This mark triggered alarms among the messengers, who would rush towards adaptive immune units, informing T helper 17 cells, those soldiers trained specifically to eliminate Candida albicans faction and restore order. But when a new faction emerged, declaring itself as Candida auris, it proved to be elusive and opportunistic. It existed in multiple forms. Two of them were fluconazole sensitive and fluconazole resistant strains. Rumors had it. A new mystery unfolded. How do dendritic cells respond to the Candida auris strains, if they recognize them at all?
An important virulence feature of Candida auris is its outer Mannan layer. This layer reduces and delays the recognition of innate immune cells, such as dendritic cells and macrophages, allowing the pathogen to survive. On this slide, we focus on the core of our study. Our research question asks whether dendritic cells respond differently to sensitive versus resistant strains of Candida auris. Here is an overview of our experimental workflow. We started with mouse dissection to obtain bone marrow, followed by isolation of dendritic cells. After incubation, we cultured Candida auris with the dendritic cells to analyze the immune response using gene expression techniques. Here are some pictures we took while dissecting the two long limbs of the mouse. And here it demonstrates that we isolated the bone marrow from the long bones and processed them to obtain stem cells. These stem cells were then cultured using FLT3 ligand, which promotes their differentiation into dendritic cells. This step is critical as it ensures we have a population of immune cells for our experiment.
To answer this mystery, a controlled encounter was established. First, the protagonists' dendritic cells were isolated and nurtured. Then, the antagonists' Candida auris strains were both cultivated into broths to allow their growth in numbers before counting them under the microscope. The encounter was held in a co-culture, each well acting as an arena, where each dendritic cell is assigned 5 Candida auris cells to recognize, engulf, process, and respond to, all during a 24-hour long incubation. After the 24-hour had passed, what remained in the wells were survivors and their weapons. Those weapons produced were molecules, some of which remained in RNA form, functioning as reports of what had happened during their dwells. Hence, RNA was looted through RNA extraction process to decode dendritic cells' decisions against Candida auris. But the issue with RNA is its unreliable nature, for it was fragile, messy in its messages, and difficult to interpret. It needed to be enforced through converting it into cDNA to preserve the valuable messages it harbored. This conversion was called cDNA synthesis. Though a durable transcript was synthesized, noise remained in these messages. Only signals needed to be archived in the shelves of strategies had to be present and even amplified to make finding them easy in these shelves. Hence, polymerase chain reaction (PCR) was chosen to be our blind scribe, copying only what it was instructed to, guided by primers, which were short sequences acting like markers, one signaling the start of the copying and another marking the end of it. Anything outside these markers was discarded. These markers were pre-decided through the familiar protocol dendritic cells followed when facing Candida albicans, acting as a reference model to how dendritic cells would respond to Candida auris. Thus, the targets were interleukin 10, interleukin 12, interleukin 1, TNF alpha, and to control the scribe, we used GAP-DH. The final product was a neat and amplified narration of what was produced, easy to read and conclude from. What was repressed through what did dendritic cells recognize Candida auris, whether they reacted differently to the two members of that faction, fluconazole resistant and fluconazole sensitive and fluconazole resistant Candida auris. What remained was a detailed gene expression analysis and interpretation of the cytokine profiles, encouraging future investigations uh, through a real-time PCR rerun or flow cytometry using different markers like CD11C, CD11B, CD86, and HLA-DR.
Sincere gratitude is extended to the University of Sharjah Research Institute for providing the resources and supportive environment that made this work possible. Appreciation is also given to Dr. Mohammed Ghaleb and Ms. Aya Tawfiq for their guidance and supervision, as well as to Tahrin Iman for her valuable contribution to the experiment. Thank you.